Bioinformatics and the Genomics seminar today. We're very glad to invite Dr. Qian Zhang from Rockefeller University to talk about her science paper. And on February 23rd, we have another speakers from Columbia University to talk about their Nature's Medicine papers. It's also COVID-19 related. So if you are interested, you are welcome to join us. And if you miss any previous seminars, we also put the YouTube link here so you can watch the our recorded seminars later. And with that, I will hand it over to Dr. Adrian Randolph from BCH, who will co-host this meeting with me and also introduce our speaker today. Thank you, Adrian.
Thank you. So we're so fortunate to have you today, Qian, to update us on your really important work in science. I also worked with Helen Su as we went over. Dr. Zhang is started off at Fudan University, getting a master's in immunology, and then went to do a postdoctoral fellowship at NIAID with Dr. Helen Su, identifying DOC8 as a error primary immunodeficiency, and then later moving on to other interests, but now working at Rockefeller University with Jean-Laurent Casanova and other collaborators on this very, very large international multi-center cohort to try to understand genetic susceptibility to COVID-19 and how it affects disease severity, because as we know, some people get very sick and other adults have minimal symptoms. So she's going to share with us the findings about type 1 interferon, and then we're going to leave quite a bit of time for questions. And please put your questions in the chat, and then at the end we can have a lot of interaction with the questions. Thank you.
Thank you very much for the invitation. Thank you very much for the introduction. I'm gonna share my slides. Okay, so thank you everyone for your time, and I'm pretty excited to talk to you about our two recent discoveries. Actually, I'm gonna talk about two stories about COVID-19. And please, if you have any questions, we have plenty of time to discuss, I think. So everyone already know this pandemic started over a year ago, has affected our lives every way possible. And this is a topic that our lab, the Casanova lab, started to work on very quickly. When this happens in March, we already started our project, because what we see in COVID-19 now is essentially something that the lab has been studying for the last 20 years. Now, everyone during COVID see that most of the people who are exposed to SARS-CoV-2 are either asymptomatic, or had very, very mild symptoms like fever for a couple of days, coughing. But then there is a minority, a small proportion of individuals who went on to develop severe pneumonia, encephalitis, or other severe symptoms. Some end up in ICU and some passed away. Now, this is not unique to COVID. This is what we see in every single infectious diseases. For example, bacterial infections, fungal infections, other viral infections. It's always the case that only a minority of individuals who get exposed to the infection develop life-threatening diseases, while the majority exposed individual had asymptomatic or very mild self-limited disease. And the lab has been studying this for 20 years. Our goal is to find out why only these minority of people had severe life-threatening infections. And we hypothesize that the inter-individual variability is caused by the genome of the host. It's genetic.
Sorry to interrupt, Qian. Can you make it full screen?
Okay. Good.
Yeah.
Great. So here is the illustration of how we consider this happen. Some people are mild or asymptomatic. Some people are life-threatening. Now, I labeled these little figures here with different colors is because not only we think that these are minority of people who carry genetic lesions that leads to the life-threatening disease, but also there are different genetic lesions or different mechanisms that all of them end up in life-threatening disease, but the reasons are different. So to study this, we established this consortium called the COVID Human Genetic Effort. This is really a outdated figure. We have over 40 centers all over the world. They can recruit patients, and they can also do sequencing and analyze data and study the mutations with us. We also have over 200 local physicians who work with us to recruit patients. And this is really a collaborative effort. And Helen Su, that Andrea just introduced, and Dr. Casanova, they co-host this consortium. And when we start to study these patients, we took a step back and started to test a very simple hypothesis. We already know that in severe, in life-threatening influenza infection, because that the patients can carry mutations in the TOR3 and IRF7 dependent interferon, type 1 interferon pathway. So here's an illustration of it. In 2015, we've discovered the first patients with IRF7 mutation that leads to life-threatening influenza pneumonia. And then we also discovered the IRF9 and the TOR3 mutation that also leads to life-threatening influenza pneumonia. And all these genes line up in the type 1 interferon pathway. I think type 1 interferons probably you're familiar, most of you are familiar with it. There are 3 types of interferons, type 1, 2, 3. Type 1 and 3 are very similar in its pathway, and type 2 is kind of by its own a separate pathway. But we found that patients with mutations in the type 1 interferon pathways are susceptible to influenza. Now, when COVID started, because it is so similar to influenza, they're both RNA viruses that mostly attack the lung. Their ways of transmission very similar. And also because the antigen drift and shift in influenza, influenza also all happens in pandemic, for example, the 1918 Spanish flu, or endemic from seasonal flu to endemic influenza. So because these 2 diseases are so similar, we want to test this hypothesis that the same mutations in the type 1 interferon pathway can also cause life-threatening COVID pneumonia. Now, the red ones are the ones we already know that can cause flu. And the blue ones are the other genes in the pathway that are already been shown to cause other life-threatening viral infections, although they have not been reported in flu patients. So there are totally 13 genes in the pathway, and we simply tested whether life-threatening COVID-19 pneumonia can also be caused by mutations in these 13 genes. And when we collected over 600 patients at that time, now we're probably over 2,000. And we also have several hundred control patients who are asymptomatic. And so we studied all the variants found in the genome of these individuals who are, have which has a minor allele frequency that's less than 1 in 1,000. So minor allele frequency is a genetic term. Essentially, it means how many, what's the percentage of people who carry these variants in the population. And we took 1 in 1,000 based on the mortality of COVID-19, which is somewhere between 0.1 to 1%. And also, because these genes are known to cause other viral infections, so they must be rare in the population. So on these 13 genes, we found totally 118 rare variants in these 13 genes in the severe COVID-19 patients. And we tested biochemically all these 118 variants. I'm not gonna go through each gene one by one, but I'm just gonna take, for example, TOR3 as an example. We test all the variants in its solitary, meaning in this case, we took a TOR3 deficient cell line, transduced different variants showing different colors here. So the red ones are the ones we found in the patients, and the blue ones are the positive control that we already know are loss of function variants. And then we transduce these cells, stimulate with poly-IC, and measure the interferon lambda, which is a type 3 interferon in this case, in these cells. And we found the one that with the little stars on the top, meaning they are significantly different from the wild type. So now we know these particular variants are loss of function or hypomorphic variants. So we did this for all the 13 genes and all the 118 variants we found. And this work is not only done in the Casanova lab, but also our collaborators who are experts in some of these genes. And totally we found 24 loss of function or severely hypomorphic variants in 23 patients. So there's one more variant in a patient because one patient actually carried a compound heterozygous variants. Now here's the list of these 23 patients. And you can see they are all different genders, different age, and came from all over the world. And I want to point your attention to these 4 particular patients because we found 2 patients with autosomal recessive IF7 mutations and 2 patients with autosomal recessive, if not 1 deficiency. So these are the key proteins in the type 1 interferon pathway, and I'm gonna talk about them in more details. So first is that I want to talk about the autosomal recessive IF7 patients. So we already know, I already explained, IF7 is the first autosomal recessive disease we found to cause life-threatening influenza pneumonia. And really to our surprise that we found 2 patients with autosomal recessive IF7 mutations, and before COVID-19, they were totally healthy. We were lucky to get blood samples from these 2 patients. So you can see patient 1 here and the patient 3 here. Patient 1 has a homozygous mutation, frameshift mutation in IF7, and patients 3 have a compound heterozygous mutation in IF7. And we got actually a lot of blood from this patient 1, and we were able to purify the plasmacytoid dendritic cells from patient 1. And I forgot to mention before that is IF7 is a transcription factor triggers the response to type 1 interferon. And it is constitutively expressed in plasmacytoid dendritic cells, which is the professional interferon type 1 interferon producer in the blood. So all the tissue, for example, your lung or other tissues do express IF7 when the cells are infected by the virus, and IF7 can induce type 1 interferon signaling. However, in PDC, IRF7 is constitutively expressed. And when PDC senses there is a viral infection, it produces interferon beta mainly and other type 1 interferons and makes the first wave of type 1 interferon response that really amplifies the antiviral effect. And now we were able to get blood samples from patient 1, which is totally, the protein is totally not expressed. And we purify the PDC from, this patient. And we infected the PDC with SARS-CoV-2 or influenza A viruses. And then we did an RNA-seq on these infected cells. So here the x-axis is the wild-type person and the y-axis is the patient. And we plot all the gene that is up or down regulated after the infection. And each dot here is a gene. And you can see that in both infections that the control makes a huge amount of type 1 and 3 interferons in responding to the viral infection. They're over 10-fold increase in the PDCs, while as in the patients, most of them are pretty much not induced or in some cases even reduced. So by doing this, we show that the patient PDC that is IRF7 deficient cannot produce type 1 and 3 interferons in responding to the viral infections. This is really to our surprise because this person who is a 49 years old woman who lives in Belgium, she was never hospitalized for any other viral infection. In fact, other infections at all before COVID-19. And we took her blood and ran a virus scan, which is looking for serology antibodies to any common viruses we know that exist in the community. And we can show that she and her family members, so she, where's my cursor? So she here and her family members and the people, we got a lot of control that age and gender matched who lives in the same community with her. Essentially, all of them are exposed to the common viral infections. That means she, as anyone else who lives in the neighborhood, are exposed to these common viruses, but she never had a problem controlling these viral infections. More specifically, we typed her serology to different strains of influenza. And again, compared to the people who in her same neighborhood, and showed that she, as everyone else, were exposed to multiple influenza A strains. That means she, as a autosomal recessive, totally loss of function IRF7 deficient person, are able to control viral influenza infections in the past 49 years. Now, I'm gonna move forward to another patient that is the autosomal recessive IFNAR1 deficient patient. So again, here is the type 1 interferon pathway, and the IFNAR1, IFNAR2, these two genes makes the receptor for the type 1 interferons. There are totally 17 different type 1 interferons. They're all very similar in terms of the protein and structure, and they share a single receptor that is IFNAR1 and 2. Now, if there is a defect in the IFNAR1 and 2, that means the cells cannot respond to any of the 17 type 1 interferons. And so we have these patients show here who carry a homozygous mutation in IFNAR1. And first we show that the patient has totally lost of expression of IFNAR1 on the cell surface. And we then stimulate the patient cells, different cell types, including the T cells and the fibroblast, and show that the patient cells are able to, were not able to respond to interferon alpha or beta. But the patient cells are totally normal in responding to interferon gamma. And show by phosphorylation and STAT1 that is downstream signal of IFNAR1 and 2. So by doing this, we showed that the patient who is autosomal recessive, if not one deficiency, cannot respond to type 1 interferon. And so I just showed you that the patient with deficiency in IF7 and if not one who cannot make type 1 interferon or cannot respond to type 1 interferon, but does that defect really leads is the cause of their COVID-19? And Jeremy Le Pen from the Cherry Rice Lab, which just got the Nobel Prize last year, showed this mechanism in a very elegant way. So what he did was he took the patient cells, fibroblasts, and transduced it with ACE2 and TMPRSS2, the two receptors for SARS-CoV-2. Now these fibroblasts, skin cells, are susceptible to SARS-CoV-2 infection. Then he stained for the spike protein intracellularly, looking for MFI and percentage to show, which is representative of the viral proliferation in the cells. So if a dot, each dot is one sample. So if the dot is on the upper right corner, meaning that there's more viral proliferation in these cells, meaning that the cells cannot control the viral infection. Now, so he first tested the patient cells with either autosomal recessive or autosomal dominant TOL3 defect and showed that the patient cells cannot control the viral infection. However, if he pre-treated the patient cells with interferon beta, the cells now can control the viral infection because TOL3 is a viral sensor. So if you pre-treat cells with interferon beta, it overcomes the defect in the viral sensor. Now the cells can respond, can control the viral infection. Then he tested the patient cells with autosomal recessive, if not IF7 mutation. And again, the patient cells are unable to control the viral infection. And if the patient cells rescued by Y-type IF7 or pre-treated with interferon beta, again, the patient cells can control the viral infection. Finally, he tested the patient cells with autosomal recessive, if not one deficiency. Again, the patient cells failed to control the viral infection. And this time, pre-treat of interferon beta does not rescue the function because the receptor is gone, but overexpress of the Y-type, if not one, rescue the function. So by doing this, he showed that the patient cells either cannot sense the viral infection or cannot produce type 1 interferon or cannot respond to type 1 interferon, are unable to control SARS-CoV-2 infection. And this, I think, is the key experiment that really established that the type 1 interferon is the key to control SARS-CoV-2 infection. And so I just talked to you about these patients with mutation, these 23 patients with mutations. At that time, it's about 3%. These 23 patients are makes about 3% in our patients with severe COVID-19 infections. And this genetic mutation really gives, how do you say that? It functions like a compass, a lighthouse to us, because even though this is only 3% of the severe patients, you know, these are very, very rare mutations, and a lot of them, the autosomal recessive diseases, happen in patients with consanguineous background. So this definitely does not explain why there are millions of people who are dying from COVID-19 all over the world, but it functions like a compass to us to point us to the right way. It pointed to us that the type 1 interferon pathway is really key to control the viral infection, although these patients themselves are very rare in the population. Now, this leads to the second part of our study, which is a discovery made by a PhD student in the lab, Paul Bester. So now I'm going to switch to slightly a different story on the other side. Now, so Paul discovered that the autoantibodies against type 1 interferon can also lead to severe COVID-19. And this is not a new idea at all. For many years, we know that autoimmune diseases are phenocopies of inborn errors of cytokine immunity. What does that mean? It means if you have a defect in a cytokine or its receptor, it will lead to the same phenotype as if you have an autoantibody against the same cytokine. So this has been shown in the history for many times. I'm going to give you 3 examples. One is inborn errors in type 2 interferon gamma, either in the cytokine itself or in the receptor, leads to mycobacteria diseases, that infections to weak mycobacteria or BCG infections or TB, and autoantibodies against interferon gamma patients also have mycobacteria infection. This has been discovered many years ago. And second example, that is mutations in IL-17 or its receptor leads to candidiasis, in fact, fungal infections. And autoantibodies to IL-17 also leads to fungal infections. And the last example is more recent, that is mutations in IL-6 leads to staphylococcal diseases. And autoantibodies to IL-6 leads to the same disease. Now, these examples tell us that autoantibodies are phenocopies of genetic mutations. So when we discovered that there are 3.5% of people with severe COVID-19 have mutations in type 1 interferon, we immediately search for autoantibodies against type 1 interferon. And even that idea was not new at all, because autoantibodies against type 1 interferon has been discovered 30 years ago. Or maybe 40 years ago. So there are several cohorts of people, patients have been discovered to carry sky-high type 1 autoantibodies against type 1 interferon in the past. First is in some patients who were treated with the cytokine interferon alpha 2b or beta, and in a small minority of women who have lupus. And in almost 100% of patients with AIR mutation. AIR is the gene that presents self-antigens in the thymus that depletes the self-reacting T cells. Almost all of them have sky-high autoantibody against type 1 interferon, and this has been discovered many years ago. However, what's interesting is these patients, although they have their own diseases, they don't present viral infections, or even though they have these autoantibodies. Only very, very rare cases has been reported that patients with these autoantibodies can actually have severe viral infections. The cases are so rare, and these patients, they already have other fundamental conditions. Was unclear what does these autoantibodies do in these patients. Looks like they're mostly clinically silent until last year, that we first in our cohort with severe COVID-19 patients, we found 3 APS1 patients who have AIR mutations. And we immediately connected dots because we already know they have autoantibodies against type 1 interferon. And so first thing we did was we screened for these autoantibodies in the patients. So over over 900 patients we screened, we found about 10% of them who carry autoantibodies against the type 1 interferon, including either alpha or omega. So there are alpha, beta, omega, kappa, epsilon, different type 1 interferons. And alpha, there are 13 different subtypes of alpha in this category. So totally 17 different interferons. And we found that about 10% have at least one autoantibody against one of them. And we found 0 patients in the asymptomatic or mild cohort carry any of these autoantibodies. And we found about 0.3% in healthy controls blood donated before 2019, and 0.3% of them carry these autoantibodies. The odds ratio is infinite. If you really have to calculate a p-value, it's highly significant. So in conclusion, we found an enrichment of autoantibodies against type 1 interferon in patients with severe COVID-19. And we also tested whether these autoantibodies are neutralizing. I'm not going to go through the details of these experiments. Essentially, we tested the neutralization by either testing the phosphorylation of step 1 or the ISG induction in these patients, and found that most of these autoantibodies are neutralizing actually at a very high titer. Oops. And finally, we also tested whether these neutralizing autoantibodies can actually cause severe SARS-CoV-2 infection. And this is again done by the Charlie Rice lab. What they did was they infect the cells with SARS-CoV-2 with or without pretreatment of interferon alpha in the presence of the patient's serums. And we found that all these neutralizing autoantibodies can block the protective effect of interferon alpha in a very, very high titer. So the red one is actually a monoclonal, commercially available monoclonal antibody against interferon alpha 2. And you can see that some of the patient's serum can neutralize the interferon alpha at higher titers than the commercially available monoclonal, meaning that they can actually block the protective effect of type 1 interferon at very, very low concentrations. And among these patients, the 10% patients we found who carry neutralizing autoantibodies, these patients are has different genetic background from all over the world. And majority of them are over 65 years old. And to our biggest surprise is that 95% of them are men. And we already know that men are more susceptible to have severe COVID-19. And this is even more, the gender bias is more severe in these patients who carry autoantibodies. And even among the, I think only 5 or 6 women who carry autoantibodies, one of them actually is an IP incontinentia pigmentosa patient. That is a disease caused by the NEMO mutations. So the patient has a biased inactivation of X chromosome. After birth, the cells that carry the mutation mutations all died. And so the patient, even though it's a female patient, they end up with only expressing 1 X chromosome in the whole body. So they're genetically in a way closer to men than women. So all these adds up, gave us a clue that these autoantibodies are more enriched in men than women, which is very surprising because if you consider autoimmune diseases, I think everyone would think that it's more common in women. And so far, we still don't know why men are more likely to have these autoantibodies, or at least more likely to suffer from COVID-19 because of these autoantibodies. So finally, to show that the type 1 interferon response is really key to control the SARS-CoV-2 infection, we did an in vivo experiment. That is, we measured the type 1 interferon in patients' blood during their COVID-19 infection. So we have a variety of different patients with different mutations, the red ones here, and also in the blue is the patients who do not have autoantibody or do not have mutation in these genes, but also have acute COVID-19 infections, and the patients with autoantibodies. And we show that patients, when during their acute COVID-19 infection, they produce a large amount of type 1 interferon in their blood, which is essentially absent, undetectable, or at very, very low level in patients with mutations or in patients with autoantibodies. This means that these two different mechanisms, mutations and autoantibodies, share the same feature, that is, they have low type 1 antibody, type 1 interferon in responding to SARS-CoV-2 infection. It also shows us that there could be a potentially treatment that this by giving them type 1 interferon. So the patients with mutations in the type 1 interferon receptor probably not going to respond to it, and the patients with autoantibodies will not respond to the type 1 interferon treatment. However, I think I mentioned before that the patients with autoantibody have autoantibody against interferon alpha and omega, very rarely have autoantibody against the interferon beta, which is another type 1 interferon. And interferon beta is commercially available as a drug that can use. So essentially, this points to us that except for the patients with mutations on the type 1 interferon receptors, all the other patients can potentially be treated with type 1 interferon, either alpha or beta, for COVID-19 infections. So finally, I think I have, I hope I have convinced you that patients with defect in the type 1 interferon pathway, either with mutations or with autoantibodies, can lead to life-threatening COVID-19. So I think this is just the beginning of our study. We definitely are expanding our search for genes outside of these 13 genes. And we are really looking into why these patients have autoantibodies. So I would like to thank everyone who participated in this project. We have many collaborators who contribute to this project. More specifically, Charlie Rice Lab, who did the SARS-CoV-2 infection for both stories, and Gigi and Helen, who contribute greatly to the consortium, to the study and recruiting patients. And I would like to invite you to visit our website for the consortium, and you can follow us on Twitter, follow our lab on Twitter. And I would like to answer any questions you have. Thank you very much.
Wow, thank you. What an amazing, this is amazing work here, and how fast you also did this, because this paper came out, you know, this is a huge number of patients to capture so quickly. And can you just describe a little bit about how you went about getting this done so quickly in a pandemic?
Yeah, this is a really like amazing experience for all of us. We knew as soon as the pandemic hit Europe first, and then US. I forgot to mention that our lab actually has two branches, one in Paris and one in New York. So as soon as I think in March, we started to recruit patients from both sides. And in March, we started this consortium and simply invited all the collaborators we've been worked with in the past 20 years, emailing them, asking them if they are willing to join us to study COVID-19. And many of them not only joined, but also invited their collaborators. So soon we have established like 30 to 40 sequencing centers all over the world, and local coordinators and local physicians. I remember my first search for the mutation was in April. So in April, we already have sequencing data rolling in our pipeline, and started to look for these mutations. And that just been an amazing experience. We have weekly meetings among the consortiums, and we discovered most of our patients around, I would say, June or July. And then we thought we really need to publish this.
So can you just talk a little bit about how are you doing all of this genotype? You said they're doing this genotyping locally and then they send it to you for interpretation, or are they interpreting it and then sending you the findings? Like, who found these variants? Was it a core team that was doing reading all of these exomes, or were they whole genomes?
So we're totally transparent within the consortium. Anyone who contributed data can see everyone else's data. So we did this by asking everyone to send our dry lab, our bioinformatics team, the raw data, and we went through a pipeline to make the calls, to make the variance calls, and then we put it on one server. So and then everyone has to sign a DTA, but after that, anyone who contributed data can essentially search on the server. We also provide a software that's developed in the lab that can allow you to just do a simple search. So any physician, if they have gene in interest or patient's interest, they can do like a refined search, say, I want to search for TOR3 in this patient. So by doing that, actually, we essentially are analyzing the data together.
Yeah, I think we
Very
have two more questions.
Yeah, so there's a question here about reports of association between interferon therapy and severe renal glomerulopathies in terms of this interferon pathway loss of function variant patients and the interferon pathway autoantibodies. This person, Dr. Majumdar, was asking about whether or not this could be seen in these patients that you found who have autoantibodies or loss of function variants. Did they have any other organ dysfunction, any other organ or history?
So this is a good question. Most of them were healthy before COVID-19. We have not found any, I would say, particular comorbidities that's associated with developing autoantibody besides L mutation and the lupus. But we are now expanding our search to more patients with a variety different of autoimmune diseases.
Okay, very good. So another question is about the patients with IRF7 deficiency. You know, John Laurentez reported IRF7 and IRF9 deficiencies in children with flu, in previously healthy children with flu over the years. And this question is, do you think that this represents a defect in the dendritic cells or an intrinsic defect in infected epithelial cells, or both? And why don't you think that these patients had severe flu?
That's a very good question. This is what we want to find out too. So when we discovered IF7 in 2015 and IF9, I think in 2017-ish or 2019, we at that time thought we would find patients with more broader viral infections with these mutations.
Mhm.
In fact, it's the opposite. We found these people who were totally healthy until 40, 50 years old, and now they had COVID-19. There is, obviously, there is incomplete penetrance in if you take a specific viral infections. Because all the genes I'm studying here are known to cause other viral infections, including herpes virus, simplex, encephalitis, herpes simplex, encephalitis, or influenza. But in these cases, they were healthy before COVID-19. Why is there incomplete penetrance? The short answer is we don't know. Maybe COVID-19 is a more virulent infection virus, or maybe COVID-19 is because all the other viral infections are common in the community. That means most of people, when they reach adulthood, they've been exposed and have antibodies against these viral infections, except for COVID-19, which is a primary infection for everyone, including adults. So this might explain some, might be related, but in brief, we don't know why these people who have autosomal recessive IF7 mutations were totally healthy before 2019.
Mhm.
Yeah.
So Dr. Janet Chow's asking about, given the variability and variant penetrance, have you investigated epigenetic changes that influence disease severity, or looked at somatic or structural variants that could predispose to severe COVID-19?
So we are looking at somatic mutations in the blood cells, essentially. That is the somatic mutations that we can reasonably test. And again, this is one of the hypotheses that why some people could have an intermediate phenotype, or they were totally healthy before COVID-19. Maybe they have a, incomplete penetrance because they have sort of the somatic or a milder mutations. And this is what we've been looking for too.
I see. And let's see, I have a question about the autoantibodies. I mean, our population is pediatric, under 18. And my question for you is, do you think there's an association with age in the development of these autoantibodies? Do you think that is it worth looking at children for autoantibodies? Or I mean, we are looking at them for primary immune deficiencies, but are they too young to have developed autoantibodies?
So I don't have the exact data. What's the plan is we are going to test 40,000 serums donated before 2019 from healthy donors with all different ages and gender from different genetic backgrounds. We expect to see that the elderly people have higher percentage of autoantibodies. I don't know the numbers for each age group because we're still testing it. So far, autoantibodies in children is just really very rare. Just for the ones that we have tested. Although if a pediatric patient has some conditions, for example, autoimmune phenotype, other autoimmune phenotypes, other diseases or familiar cases with autoimmune diseases, I think it's still worth testing. Mm. But in general, let's say a totally healthy child before the age of 20, the likelihood of that child has autoantibody is just very, very low.
Mm-hmm.
Yeah.
Okay, very interesting. let's see. As far as these other things on this cohort, the ones that got very sick, were these ones with the genetic variants? Did they have other risk factors as well, like obesity, hypertension, diabetes, all the other common risk factors?
Yeah, so they do. So we tried to recruit patients who have no comorbidities and relatively younger, relatively healthy, because we don't want to sequence 5,000 patients and discover the gene that causes hypertension. But in fact, you know, when after all, when we had recruited these patients, a lot of them have a variety of different comorbidities. And we didn't find any association with comorbidity with the severity of disease with or without the mutations. So I think we found that the key defect is the type 1 interferon defect. With or without comorbidities, probably going to change the course of their disease, but that does not decide if they get severe COVID-19 or not, if they get sent to ICU or not.
Mm-hmm. So your definition of severe COVID-19 was that they got sent to the ICU. Is that, and then, you know, in the elderly, the mortality rate in COVID-19 is very high. What was the mortality rate in this cohort, and did the ones with genetic primary immune deficiencies have a higher mortality rate?
So I think in the patients with mutations, we found 2 are deceased. And in the patients with autoantibody, I remember it was about 1/3 to 1/2, between 1/3 that actually died. I cannot give you the comparison to the general mortality because these patients are selected. For the patients with, so we selected, actually we tried to get patients who are younger than 50 without comorbidity, without any, you know, pre-existing comorbidity, all these different factors. So I cannot tell you how you compare that, you know, to a general population. I think as we are now expanding our recruitment, we are now recruiting patients from all ages and all different conditions. We will get this number more precisely in the future.
Okay, thank you. Are there other questions here from anyone? Please put them in the chat or feel free to ask. If not,
I just want to say that there's one researcher from Dr. Qian. She has to leave, but she just wanted me to tell you that this is the one of the best talk she has ever heard. Thank you for the great talk.
Thank you. Thank you very much.
So what's on the horizon for your group? You mentioned a few things, but that you're expanding across the ages, you're expanding the interferon variants. But what are there other things that you're working on across, and has your recruitment continued to rise with this second wave of COVID-19?
Yeah, unfortunately, our cohort has expanded really dramatically over the last several months. We are now looking, so when we started, we picked the most extreme phenotype, that is like the totally asymptomatic and the most severe one who got intubated on ECMO. Now we are now recruiting also patients in between. So the moderate or the relatively severe cases, or the, you know, these cases. So we, that is one thing on the side of recruitment, and we are expecting to see maybe milder defects or lower titers of autoantibodies in this cohort. We also, on the autoantibody side, we're testing 40,000 normal control. That would give us the real odds ratio and the percentage of people who are autoantibody positive. It will tell us if this is age related or actually if it's truly gender related. And we, on the mutation side, we expand our search to genes that's not specifically in the type 1 interferon pathways, but there are hundreds of ISGs. That's the genes that induce the by type 1 interferon signaling, and they are actually the effectors, the protein that actually carry out the function to kill the virus. And we're focusing our search in these genes because they might point us to a therapeutic target, because these are the molecules that can kill the virus. So we're really focused our search on these genes. And I think we're also expanding our consortium to reach out to more countries and more regions.
That's fantastic. Does anyone else have any questions? This was really, really amazing talk and amazing work.
Thank you very much.
Thank you so much for sharing it with us. And if there aren't any further questions, we'll close. But thank you so much. This was really helpful.
Thank you. If you have anything, you can email me or follow us on Twitter and we can discuss. Thank you.
Thank you.
Thank you. Bye.